You are listening to Neurosalience, the OHBM podcast.
Welcome to the OHBM Neurosalience podcast. In this podcast today, in this episode, this is our sixth podcast so far in our series. We're going to be talking to Chen and Molly Bright. Jean is from the University of Toronto. Molly is from Northwestern University. And they both, all their careers have been exploring and developing methods to extract physiologic information from fMRI time series, as well as with MRI as well. In this podcast, we're going to talk about some of their work. We're going to have like a big picture on how it's relevant to fMRI, how it's relevant to clinical issues, how it could be used clinically. And maybe we'll dig in a little bit into some of the details of some of their papers, some of the more interesting things that I've found from reviewing what they've done. And the second part of this podcast actually is unique in the sense that, they're both women scientists, and they both really wanted to share their experience of being women scientists and offering potential solutions to the unique challenges that women scientists have all the way from undergraduate up to their level of being PIs of labs. So really, really interesting conversation all the way through. So I hope you enjoy it. Thanks. So Jean Chen received her master's in science in 2004 in electrical engineering from the University of Calgary and her PhD in 2009 in biomedical engineering from McGill University. She completed her postdoctoral work on multimodal MRI and brain aging at the Martino Center for Biomedical Imaging in Harvard Medical School in 2011, and then joined the University of Toronto Medical Biophysics program as a faculty. She's a senior scientist at the Rotman Research Institute and a Tier 2 Canada Research Chair in neuroimaging of aging. And Molly Bright received her bachelor's in physics from MIT in 2006 and received her doctorate of philosophy from University of Oxford in 2011 as part of the collaboration with the NIH. Her co-mentors were Peter Jezzard and Jeff Dunn at the NIH. So Peter Jezzard's at Oxford and Jeff Dunn's at NIH. She completed her postdoctoral training at Cardiff University Brain Research Imaging Center, Kubrick it's called. Then she moved to Nottingham as an independent Anne McLaren Fellow to develop ultra-high MR imaging methods for studying cerebral physiology and neurological diseases at the Sir Peter Mansfield Imaging Center. And then finally in 2018, she returned home to America to lead the Applied Neurovascular Imaging Lab at Northwestern University. And so, both of you actually have a little bit of overlap with me. I did my postdoc at MGH, so didn't overlap with Jean at the time. I did it back in the late 90s. And Molly, I saw at the NIH, when she was doing her graduate work, and then she worked with Kevin Murphy, who was a person in my lab. So we have a lot of overlap, or some overlap. And also physiologic MRI is something that's sort of near and dear to my heart. I really get excited when I see good papers sort of pulling out new type of information out of functional MRI data or even MRI data. So just to introduce both of you, thanks both for doing this. And whoever wants to jump in first, how you got started and how you got interested in this particular aspect of functional MRI and MRI. So maybe Jean, if you can go first.
Sure. Yes, thank you, Peter, for inviting me. I'm very delighted to be here. So I did an engineering undergrad. So towards the end of my degree, I realized that I was really, really interested in signal processing. All the signal processing courses were my favorites. And afterwards, I actually got into industry. I did a year of work in signal processing. At that time, I had two options. One of my options was to work on GPS, satellite signaling. And the other one was to work on biomedical research. And one of my longtime profs in undergrad encouraged me to go into MRI. So that's what I did. I thought that I would make a bigger difference in this field with my skills because I see there's, at that point, a general lack of signal processing development in medical imaging. And so in my master's, I did perfusion of stroke using MRI And then that's when I got interested in the physics aspects of MRI. So, and then I moved on to McGill to do a PhD in a sort of physics-oriented lab, and with Bruce Pike as my PI. And that's when I got my first exposure sort of formally to fMRI. And everything just followed. The project was challenging, but it was very interesting. It taught me a lot. And so I took that with me to the Martino Center, where I work with David Salat on brain aging. And there I got my first exposure to resting state fMRI. And I realized that all the calibrated bulb theory and the models that we use, and they had not translated into resting states. So I thought that was sort of like a gold mine of information and so much to be done. So that's where I took it off when I became a PI.
Okay. Without a doubt, it seems like fMRI is still catching up in terms of signal processing and applying all these methods to fMRI data. It's still, there's so much kind of, I hate to use the cliche, but low-hanging fruit in terms of that, in that sense. But so Molly, how did you get started, and what made you interested in this fMRI, this aspect of fMRI?
Yeah, thanks for having me on. This is a lot of fun, I think. I was trained in physics, and I had done a fair bit of optical imaging in various labs along the way. And when I moved into grad school, I think the only thing that kind of guided those directions was that I wanted to become a little bit more applied and a little bit more clinical and translational. And then this opportunity at the NIH and these partnership programs that the NIH has with different universities, that suddenly became a new opportunity in front of me. And it was a really great chance to start to take some of the physics and technical training and think about how they can be useful. And for me, like building a career where there are just so many possible applications where I think maybe if we go down that route, we'll make a difference. Like I like having those types of long-term benefits to things that I'm tinkering with in the short term. So that I gravitated towards imaging and medical imaging for that reason, I think. And then once I started in MRI, I think there was just a lot of random chance involved. I don't think I'm the only one that gets a bit kind of knocked around by, oh, there's this paper. I know you try something, and then it snowballs a bit. For me, I think a paper had come out where they used a breath hold in an experiment. And my mentor saw that, and he suggested that maybe as I just get my hands a little bit dirty with some scanning, I could try sticking one of those in and see what we get. And I really liked it because I didn't have to fight the data in order to see something. It was just so visible, such a robust effect. You could interpret it much more directly. And I really liked that. So yeah, I pursued it. And it's been influenced by some work in more neuroscience driven labs, psychology driven labs, physics driven labs, but this world of more physiologic imaging kind of can connect to all of those pretty naturally. So it's been a fun kind of meandering journey so far.
Yeah, no, that's exactly. I mean, it's funny. I come from a physics background, but it seems like the people who are really pushing the field are the ones who come from more the technical backgrounds, who are trying to apply this sort of thing. And as far as and also a lot of physicists sort of want to make their work, they want to make it feel more relevant, and so they get into MRI and then clinical. And it's definitely, there's immediate payoff. I follow as much as I can both of your work and then in the work of others who are doing physiologic MRI, and I find it really fascinating. And it seems like there's so many gems of information and insight from your work that while a handful or larger group of people know, it doesn't really translate to the world of cognitive neuroscience. I mean, you have cognitive neuroscientists who are super sophisticated in the processing, but then they don't realize, this bold signal is larger because there's, large veins in this voxel, and it doesn't have much to do with neural activity. And not a lot of people, I think, well, there's a lot. I mean, that said, there's always exceptions. I mean, there's many great neuroscientists who understand this. But so what are your thoughts on that, either of you, as far as, do you feel frustrated, or do you feel like there's some inroads here and there, or in what sense is there? So
so I think about this a fair bit. I think that one issue that we may have is that the people working on the physiological side, trying to just wrap our heads around what these signals are telling us and what they really mean and how that all breaks down when your assumptions don't hold. And we can often end up writing papers that could be interpreted as saying bold fMRI doesn't work. It's not a fit tool for the purpose you want it to fit, right? You can't study neural activity with fMRI because it's all about the plumbing. And I think what we need to be careful is I don't think that the richness of information on the physiological side means you can't use it to study neural activity. It just means you have to be a little bit more conscious and creative and thorough with what you collect and how you analyze it and what you ultimately interpret. But because we often have people working in one silo or another, I think sometimes our work can seem like, it's a bit of a downer. It's like, oh, well, you can't use this method for what you want. And I don't think that's actually the core message that, the physiological imagers are really internalizing ourselves. I think we just see it as added richness to the data that you should be considering, but we can absolutely try to keep developing this tool for cognitive neuroscience as well. So I'm trying to now write papers that aren't just about identifying problems, but also about, well, what can you do with what we now know? And that's a much harder set of papers to write. It's a harder research direction to go in, but I'm optimistic. I think more and more people I meet from sort of the other side of the aisle are engaged with this side of the data.
Yeah, that's why I really like your work, Molly, because you try to make the measurements more practical. I try to come from the other perspective, and I feel the frustration of the neuroscientists as they want to try to apply our findings, but it's just too hard. You have to get 3 extra measures, which you don't have time for, or you have to acquire some additional instruments, and then the resulting measurement is not as repeatable, it's not as sensitive, and so then where do they land with that? So I can feel that because these are my colleagues that I work with regularly. So for example, with this resting state vascular reactivity sort of approach, I find that to have much more uptake because it's easy, you get something for nothing. And so this is something that I think this type of technique is what I think we as physicists or as the technical crowd, we need to think more about developing for there to be uptake. Of course, it's taken a long time for anything to penetrate into the cognitive neuroscience world, but I think it's not because they don't want to transition or they don't want to hear about all the caveats. It's just that in the end, what can they do? I think a lot of us I agree with Molly also, we're embracing this silo thinking. I'm a technical person, so I only want to understand. I don't think about how to translate. I think there's a big gap there, and there's lots of opportunities for those who are in that position to translate these techniques.
Yeah, that's interesting. I think that I try to, when I talk to the neuroscientists, that you can tailor questions that sort of don't really depend on, some of the variability that you see. And so you can kind of look beyond it. But it's funny, and like both of you said, there's opportunities, I think, for, and we'll get into this, to sort of normalize the signal a little bit better so that you can actually get more quantitative information. But let me start out with a general question. So just to maybe both of you off the top of your heads. So as far as, we can extract neural information. So off the top of your heads, what other physiologic information can be extracted from the fMRI time series? Just, with any manipulation you can do, what do you think can be extracted? And then what, maybe to go into that, what is useful, what's potentially useful clinically?
Wow, that's a pretty loaded question. Maybe I can start on this one. Yeah, so I spent my whole PhD trying to extract this type of information. Oxygen metabolism, that's always a big one, the holy grail for some time. And of course, reactivity is another one of my passions, as is Molly's. So vascular reactivity, especially because of the recent advent of this huge interest in the vascular hypothesis for Alzheimer's, there's more and more attention sort of on the vascular side of things, and then really think that can take off, and I think that's important, and that's just there. That information has been sitting there, irrespective of whether we use a breath hold. That information is there, not only in the amplitude of the signal, but also in the timing of the signal, right? So there are multiple dimensions that we can exploit. And as to what can be useful clinically, I think well, while translation takes time, I think in Toronto, for example, we have a fairly strong group at Toronto Western that translates vascular reactivity measurements into the clinic in looking at patients with vascular pathologies. And so I think that's a good model, perhaps for others to adopt when possible, because it's all about the feasibility of the technique and also the commitment of the researcher, the clinical researchers and the clinicians. Yeah, so the lowest hanging fruit will probably, in my mind, be vascular reactivity, although we are working actively still on trying to extract the oxidative metabolism and other things related to the vasculature, but that will take a little bit more time.
Yeah, I think you said it very well, Jean. And yes, personally, I'm also rooting for vascular reactivity as maybe the most accessible clinical marker for translation. I think it takes very, very brave and special people to kind of bridge this gap of the developmental side of the method and really being willing to try to scale these methods up for a proper clinical trial. And that's starting to happen with this vascular reactivity mapping, trying to help with diagnosis and planning for surgical interventions. And so that is just starting now. And so I think in the next 10 years, it's going to maybe be something that you could order, for a patient and get a CVR scan done. And so that's I think our best hope at the moment in terms of real clinical take up of these methods.
So with regard to vascular reactivity, just really quickly, just as far as clinically, is it, so I mean, obviously it's maybe relevant to vessel stiffness or just general vascular patency as far as, likelihood for stroke. I mean, what do you think what patients would be, would you run a test for vascular reactivity with?
Yeah, right now I think it's perhaps most useful in patients where you think that we're approaching this risk of maxing out the dilatory capacity of vessels. So that can happen for a range of reasons, but often if you start to become more at risk for an ischemic stroke, your vessels are dilating to try to compensate for some decrease in blood supply. And at some point they can't dilate any further. And that's when real damage starts to accrue. So using vascular reactivity scans to track and try to estimate where are you in that process of, I can totally compensate for little fluctuations in my blood supply, to I can't, I'm right at the threshold. Any fluctuations might cause real damage to my tissue. And so CVR scans can help you try to estimate where you are, and that's going to vary around the brain and it'll vary with an intervention. So that's maybe where I'll see it happen first. But I think Jean mentioned the vascular framework for thinking about different dementias. And that is a slightly different vascular impairment here, but CVR could also play a role. I don't think we have a handle on exactly what that looks like yet, but that will be another avenue.
Okay. And also, it just brought to mind, other work. I mean, I think that both of you looked at this, just looking at not only the vascular reactivity with physiologic stresses, but also the lag or the relative delay. It seems that, and I've seen work, also one of you, collaborated with Blaise Frederick as far as, using a photodiode or else looking at the relative delay with the global fluctuations and, having data that compares really well with like gadolinium injections and things like that actually look at areas of compromised flow nicely just by slow arrival time. And so yeah, I mean, okay. All right. Well, good. So what do you think would be useful in terms of this information? Definitely for quantitative fMRI, it's nice to have, these M maps, as they're called, sort of maps of potential bold. How else do you think this could be useful to, I mean, I know Mala, you had a recent paper on doing these breathing manipulations at the beginning of every fMRI scan. So how do you think it could be useful to fMRI data and interpretation?
It depends what lens you want to look at your data with. So yeah, so the paper you're referring to just got out on bioRxiv, and it's something I've kind of thought might make sense for a while, and then I needed a clever postdoc to actually figure out if it makes sense and what it looks like. But she's just done that. So Rachel Sticklin just put a paper out on bioRxiv, and we're proposing this small little very practical thing that pretty much anyone could do with an fMRI scan, where you just change your breathing a little bit in the beginning to access some of these vascular measures we're talking about. So that's the dilatory capability of the vessels, and also this lag, this latency of the hemodynamic response. So you get information about the health of the vasculature, which if you're scanning for any reason, if you're scanning somebody who's had a stroke or has some sort of perhaps dementia or so many different pathologies have a vascular element. So now you're able to characterize that. But if you look at the lens of the rest of the data set, your typical resting state fMRI scan, we're still hypothesizing, but I think that there's a lot that information can tell you. So you could potentially use the dilatory response, this CVR measure, or this M map to try to normalize the bold fluctuations you see in your normal fMRI data to account for some of that physiological variability throughout the brain. And then also this lag measure that we get. If you want to look at relationships between two signals in disparate regions of the brain, if there are differences in just the transit times and the dynamics of blood flow in those regions, you want to account for that if you actually want to look for true relationships in maybe neural processes. So what we're hoping to do is kind of make this hybrid approach where you can combine the information. You have this richer, more complementary data set that lets you kind of view both sides with a bit more context.
Yeah. Okay. Yeah, I mean, there's so many directions to go with this. So my ultimate goal, one of my goals, and I say it very boldly in my lab reviews, is that I think that we can remove all non-neural physiologic noise. Maybe not thermal, maybe thermal a little bit, but I mean, there's some new techniques coming up. But I think that we can get rid of all this physiologic noise if we just model it well enough. And but then Molly, you had an interesting paper that sort of suggests, just using modeling and regressing might overfit and maybe isn't the best approach. So you might want to maybe talk about that briefly.
Sure. Yeah, this is a fraught scenario. I think my first caveat here is I am not a statistician, and I thoroughly recommend that anybody who's working in these areas befriend a statistician who understands neuroimaging data, because we often use stats a little bit loosely to interpret and make conclusions out of what we see. And as I've learned, that's not as straightforward as I initially thought. So find a friendly statistician and get them on board and learn from them. But so the paper I think you're referring to, I was showing that, I agree that I really like to have a forward model of all of these sources of signal variance. So we record things about a person. We try to estimate their physiology from a nasal cannula, from a pulse ox, from a belt around their diaphragm, from optical recordings, whatever we can get. And then we try to model what those signals look like in a bold fMRI time series. And we make a big old general linear model, and we can regress out any associated variance. And then we have cleaned data. And this is how I prefer to operate, but there are serious limitations to this. And some things I really suspect it's just a matter of time. We'll get more and more clever and we'll figure it out. And some things I'm a little bit uncertain about, like, are we actually going to be able to do this? So one thing that's come up is that like this model of how something influences the bold signal. We're talking about how vascular physiology differs throughout the brain and person to person. So modeling what you expect to see from a given noise source might be cohort specific, person specific, brain region specific, voxel specific. And so maintaining a forward model of what you predict to see gets increasingly more difficult, especially when we're bringing in pathology. So people are doing this. They're working very hard to do this. Sometimes you need big data, and then you can learn from that to bring it back down to an individual subject. So I think it's in the realm of possibility, but it needs a lot more work. Yeah.
Yeah, and it seems like things vary on a voxel-wise basis too. It's like, you can't just have one regressor, and the shape varies, and all kinds of things vary as far as that's concerned. Um, it just seems that if you can model it completely on a voxel-wise, come up with some way of getting all that information quickly and efficiently on a voxel-wise basis, because it varies from subject to subject as well, and it could
and we're ignoring interactions, which is maybe my favorite thing to think about, which is none of these processes happen in isolation. Physiology is ridiculously correlated with itself. So anything you measure from one physiological source, be it neural, vascular, it's probably related to every other thing you measure. So fully differentiating is non-trivial.
Yeah, and before I get into some questions to Jean, I just want to mention this one other paper. I figure since we're talking about that, this one other paper that seems to suggest. I remember one time, a long time ago, Rasmus Byrne, who was in my group, and I had this paper that, we tried to look at resting state, the breathing response function, and we found that bellows sort of response, the spatial map looked a lot like the default mode network, and that seems to, you've taken this much further with looking at, it seems that, and it's really a weird question. It seems that there's overlap between there's respiratory signals as you get into time series and different networks in the brain. It seems like each network has their own sort of vascular network that sort of corresponds to a neural network. And so, if you're looking at these networks, if you try to regress all the respiration, you might get rid of, throw out the baby with the bathwater in some sense.
Yeah. And this, I think the paper you're referring to it's my favorite one to think about. And it's the one I really don't know how to follow up. So please, anyone listening, if you have great ideas, please get in touch, because I think it's a really just neat puzzle to work out of. So what we did is we had, in one experiment, we had some of these vascular stimuli, and then we had your typical neural stimuli as well. And we averaged a lot of data together from different people in different sessions, and we just looked for structures. And what we saw were, say, two networks. Both of them kind of look like the default mode network, but one of them looks like when you look at the time series of these networks, one looks like a working memory task, and the other one, with very similar spatial features, looks a lot like our vascular stimulus. And we saw that for a visual system, and we saw that for the sort of task positive network blobs. So the working hypothesis that we were playing with, which we can't prove with fMRI or with the data at hand, but the hypothesis is that, it's not unreasonable to think that our vasculature and our physiology has developed in tandem with all of our neural systems. So there might be some very network specific physiological processes. So yeah, teasing apart these systems, maybe that actually isn't what we should be aiming to do. We just need to make sure we're characterizing sort of both processes and both systems in order to interpret something in a given network. But yes, I think we're going to need multimodal imaging. We're going to need many more clever people coming to this question. So if this intrigues you, please get in touch, because I think it's a neat thing to think about.
Yeah, no, without a doubt. I think, it's like anything in science, the story gets a little bit more complicated. Yeah, there's more questions that arise as you dig in, and you're like, oh, this isn't as simple as we thought. So Jean, speaking of multimodal, so I saw in your lab webpage that you work with EEG and fMRI. So how do you apply that as far as with what you're working with?
Well, yeah, so I only got into EEG quite recently. And this is, again, a suggestion to all those out there. If you think you're an MRI person, don't, because you just never know. Because as I got deeper and deeper into resting state, fMRI, we were just busy trying to tease apart the supposedly neural and the non-neural components of the signal. And I got the sense that a lot of the signal, actually in resting state, maybe the majority of the signal may not be neural. And so that's when I felt it was absolutely necessary to bring something else. Of course, EEG is not perfect. It's not the silver bullet. It has its own challenges, but it gives a different perspective. And so it's necessary. And people say that bringing multimodalities into your work doesn't only give you the benefits of both, it also gives you the challenges of both. But I think this is a direction that I've enjoyed taking. For example, if we were looking at the physiological processes. For example, Rasmus's RVP, so respiratory variability measure, we assume that is not neural, but is that true? So I think one layer of this challenge is that we don't know the interactions between neuronal processes and the physiological processes. And I felt like I was just starting in this, and there are people in the field, not particularly in our field of research, that have probably been looking at this question for decades. And I think this is a golden opportunity to kind of reach out to those experts who may not be in MRI, just to get a better sense, because we don't want to be overly naive in thinking. I mean, I was personally, I think I was naive in thinking that we need to throw out the physiological processes because of the literature that I've followed. But this EEG experiment, which just came out in Neuroimage this year, is changing my mind. It makes me realize just how much I don't know. And so on the one hand, these physiological processes might be involved in a feedback loop with neuroactivity at both low level and high level. And they interact with each other. So it is a messy problem trying to really get down to the neural, because then at this point I thought I was onto something, but then when I get there, I realize it is not what I thought it was. So
yeah, I mean, both of you, as you're talking, it brings to mind, this work by, I think it's Greg Moore. He was at MIT. Was at MIT. I'm not sure exactly. And yeah, he actually had this hemoneuro hypothesis, where the hemodynamics actually triggered neuroactivity, which, and I think he pursued it, but I think that everyone hated it. And
it's an amazing paper, though.
It's a really cool paper.
It's just saying the vasculature passes the Turing test. So just because it's not what you think is driving some of the aspects of function and cognition doesn't mean it isn't a powerful system that maybe could have impact that we don't yet understand. So it's a really great read.
Yeah. And I think that there's still work to be done with that. I don't know if he's still doing specifically that work, but it was beautiful. It was a beautiful paper. So also, Jane, and speaking of beautiful papers, I was just looking at, I learned something that in your paper characterizing the transit time of blood throughout the entire brain using, I think, the myconnectome data set. That was really cool. And it's like something like 7 seconds, and you characterized it from, all the way from the jugulars all the way down to, or not the jugulars, but the arteries all the way through.
Yeah, through the sagittal sinuses. Yeah, so I was brought onto this work kind of late. The idea was really from Yunjie and Blaise. And so there, as it turns out, there was this reproducible anti-correlation between an artery and a vein. So in this case, it's a major artery, in the internal carotid, and a major vein. And we know their orientations. And so it was highly reproducible. It came out that the lag between them was about 7 seconds. And so the first implication was that you might be able to use this property to map transit delay elsewhere in the brain. So at that point, the question to me was, so from the perspective of MR physics, how does that happen? So we have simulated this phenomenon, actually, from first principles, susceptibility, and the equations laid out by Ogawa and al. In 1993, really seminal paper. And we find that indeed, based on susceptibility effects alone, there is always going to be an anti-correlation between arteries and veins, especially in resting state. When you're looking at correlations, perhaps the arterial signal itself is very small, but if you're looking at correlations, it might still be very robust and reproducible. But we also further this investigation into other vessels in the brain, so where susceptibility can still be modeled, but there is this more complex interplay between vessel orientation and occupancy in the voxel. So this work, I'm hoping will present at the OHBM this year. I mean, we've found something very interesting, but I think this is also not something very hard to do. I think all physicists can do this, but it's just, something that's sitting there and no one has really looked at before, Blaise and his lab.
Yeah, actually, to be honest, it kind of, it's funny. It kind of blew my mind when I read, because I always assumed, I studied those papers a lot, and I never worked it through that the arterial susceptibility is actually different from tissue, and so it can give a bold effect, the opposite sign. And there's so much interesting information that could just be obtained from that in itself. And it changes sign right around 86% oxygenation. That's really, really interesting. So
but in task-based experiments, this could also apply because the arterial signal is also driven by T2 and T1. So these things become drastically longer. T1 becomes a lot longer at high field and T2 a lot shorter. So the difference between arterial T2 and tissue T2 also becomes larger at high field. So I expect, although I've never worked with high field, this effect might be enhanced.
That's very cool. I'm definitely going to think about that for my own lab's work, and potentially. I mean, there's so many interesting. I could talk for hours, but so why don't, just so that we have enough time to talk about our other topic. But what do you think? So just to conclude this part. So what do you think the long-term prospects are for this research? I'm always trying to, even talk to the people at the NIH who, give funding or whatever, just to say, hey, this is really important research. I personally think that the first clinical applications of fMRI will be things like this, rather than coming up with neuropsychiatric biomarkers or whatever. That are, I think it's, and so what do you think? What do you think are the, you already hinted at the biomarkers that might be used as far as vascular reactivity, and what information do you think is remaining maybe to be extracted? And what do you think the long-term prospects, like in the next 10 years, are for this research and maybe the applications?
Yeah, so one thing that I guess we haven't touched on very much so far is the idea of looking at autoregulation in some of these same signals. So cerebrovascular reactivity is typically the response induced by something like carbon dioxide. And that is going to be different local physiologic mechanisms to your autoregulation response, which is so critical at maintaining blood pressure and supply to your brain throughout your daily activities. So it is getting at a different process. And I think from a clinical point of view, the idea of measuring autoregulation with some of these techniques would be a huge leap forward. It's tricky. Again, we don't, to do a blood pressure challenge in the scanner. When I was in Cardiff, we tried to do this. We built what we fondly refer to as a leg coffin. It's a big wooden box that you put your lower part of your body into, and there's a vacuum cleaner in the control room connected, and you basically just reduce the pressure in the box, and blood pools in the legs. And you can do some of these types of experiments, trying to understand how signals in the brain related to physiology change with blood pressure.
Can you squeeze the box too? Can you?
Yeah, there are anti-shock trousers which actually cause positive pressure. We didn't try those. It's hard, right? We sucked people into a box, so their head moved several inches out of the head coil. And when you're doing some of these very sensitive scans, that type of movement, it's just very destructive. So we had to kind of make this work, but. So maybe not that experiment, but ways of accessing blood pressure recordings in the scanner. There are some beat-to-beat recordings we could get to maybe pair those recordings with fMRI-like acquisitions to try to get information that right now we only have Doppler readings. And then we could really look at autoregulation and map these things. And that'll play a huge clinical role if we can really tack that, I think.
Yeah, I often thought that just simply having continuous measures of blood pressure and in which you can, just like along like, both of you have, maybe Molly does more breathing tasks and Jean maybe looks at resting state variations in end-tidal CO2. But the same thing in blood pressure, you could look at maybe blood pressure tasks, or maybe, I don't even know how much blood pressure varies over time and continuously measuring it, just using that signal to.
Yeah. Yeah, it's always a trade-off, right? If something that's easier to acquire often has a risk of you don't have enough variation to really powerfully see the effect. So I think in the beginning, it's helpful to modulate things until you can pin down models and you know what you're looking for. Simple things like a thigh cuff release are being used, and that's a way to have this big pressure change that's very transient. So there are things that we can do, but yes, having continuous recording that's MRI safe and really is getting at a blood pressure recording, I think we're not quite there yet, but there are things in progress.
Okay. So Jean, what do you think in 10 years? What do you think?
Technically or clinically?
Maybe a little bit of both. Maybe a little bit
Right.
of both.
Okay, so in terms of clinical, maybe this is closer to where we are right now. I've kind of stuck to my guns and said we don't want to introduce any breathing challenges, although they're not, outright breath holds or respirac sessions, but I mean, you need some sort of variation to get a robust measurement. So that has been a real signal processing challenge, and but I see the enthusiasm in the users, and they want to use this technique. So I think this is something that we need to work on. We need to push into the clinical realm in the next 10 years for sure. But you mentioned blood flow, blood pressure. So blood pressure is a Pandora's box. I find it has so many implications. We know older adults tend to have higher blood pressure and higher vascular stiffness, also linked to risks for various diseases, including Alzheimer's disease, other forms of dementia, diabetes, you name it. So this has a broad implication to a large portion of the population. But then it has other physiological feedback mechanisms. So I'm reading into this, and it's become like a new obsession. Also, vasomotion is another thing that's been really puzzling, and we're just trying to sink our teeth into it. And the more we know, the more we don't know. So with that, I'm kind of hoping that vasomotion can be a clinically useful measurement wherever it comes from. It's there. I think we try to capture it. It's very evasive, but we try to capture it, and I think it's also very elegant in the sense that it is in the data. It can be tracked by its frequency and so on.
Yeah. Yeah, you actually broke down. So in one of your papers, I'm sorry to go back into the weeds, but you have another paper that breaks down the main modes of the resting state, and one of them you identify as vasomotion. I thought that was really interesting because it's a big problem in interpreting, what is vasomotion, what's not. I mean, everyone thought that the frequencies kind of overlap, but you're able to separate them in that regard.
Well, as we thought we were, I think it's never the end of the battle. But it was one approach. Which we thought we did our best, because I was very determined to tease out the vasomotion. So I mean, I don't know that we have a full understanding of it. So I know the Kleinfeld lab has been actively working on this, showing that it's got implications neuronally of the gamma activity, whereas in other literature it's regarded as mostly a systemic oscillation. And when you look up the definition of vasomotion, there's confusion with vasomotion versus the Mayer wave. So this is again, so we have a paper that's coming out in Human Brain Mapping soon, in which we sort of investigate the presence of this wave in the brain by using the signal from the cerebrospinal fluid. So it's sort of a first try, but I think that's something that's also we're going to pursue. So coming back to your question about the 10-year sort of plan, or where I foresee the field in 10 years, I hope that from the technical side, we achieve sort of a more unification of calibrated fMRI between task and resting state. This is what we're also working on right now. I believe, coming from a background of gas calibration and breathing challenges, I actually have become really fond of the gas-free approach. Not only because, I mean, it's more math and I like that, but also I recall when I was a postdoc at the Martino Center all those years ago, I had this chat with Bruce Rosen, who said that I should, despite the popularity of resting state, get down to the basics. I mean, read your paper from 1995, read Jared Boxman's papers. And that's what I did. I was in the reading room reading those papers, and I thought, this is very cool. And that's when I got into sort of quantitative bold, more quantitative bold. I think we all need more quantitativeness in these measurements. Otherwise, I mean, it's in 10 years, we'll still be as frustrated as we are now.
Yeah.
So from the physics perspective, I hope that we're going to achieve some sort of calibration that applies to both task and resting state, to get more information from that and about the vasculature. And to do that, I think we need to model the signal. So this is,
Yep.
You did this so many years ago. I mean, I don't mean to imply that you're old, but it's just something that I was pretty impressed with. And we still haven't seen broad sort of application of this type of approach. Frankly, I really love it, and we're getting into it now. And I think using this type of approach, we have more computational demands. I know that Boxman simulations take a long time, but it doesn't have to anymore because we have machine learning, we have clusters, and so we can model a lot more things than we could before.
Yeah.
And by doing that, we can also extract a lot more parameters, things that we don't usually get access to. So quantitative parameters, I hope to see in 10 years.
That's a great answer. And I think you're right. I think, we haven't even mentioned machine learning sort of being more efficient and pulling all this out. I'm trying to talk to people along those lines too, in terms of denoising and whatever, but still pulling out the physiologic information. So just to, we're going a little long, but if you don't mind, we'll go a little bit long just to make sure we have enough time to talk about the other part of this. But maybe in the context of women in science. So what has been your experience going from doing, your undergraduate work all the way to being now starting your labs as women in sciences? Where have you seen blocks or frustrations, and are there any potential solutions to that? So I'm curious.
So I guess, a cheerful way to start this is probably Molly and I were amongst a group of female PIs that gathered at one of the meetings that was held in Baltimore. And then we just naturally clustered and we started to chat about our experiences. And it was relaxing and supportive environment. I think personally for me, coming out of a lab, being a student, becoming a PI, definitely has exposed me to things that I just never learned in grad school. Things like how to navigate the department, all the services that we have to do, expectations from students, expectations from colleagues, and frankly, drawing a line in terms of the boundaries in our responsibilities, especially now during COVID, where I read this article recently, basically summarizing how women typically have more caregiving responsibilities, and they have more service and teaching responsibilities at the university. So these are things that are not just specific to us, but also to others. And so basically how to navigate that, I think, is something very new. And I could feel that sort of supportive environment that we had. Molly and I were at this dinner. I wish others had it too, so they could share their experiences and support each other. Maybe there are no solutions immediately to some of these issues, but at least it really helps to know that you're not alone.
Yeah, and I think when Jean and I have talked about this previously, thinking about the benefits of mentoring. And so this was an example that she's talking about of, I guess, like near peer mentoring, of people who are kind of in the trenches at the same point and with the same types of experiences that we're going through simultaneously. And then it's also very helpful to have more senior colleagues as mentors. And until we have excellent diversity at all career levels, you have to make an extra effort to find and connect with diversity in senior colleagues as mentors. So, I mean, I think I've been very fortunate. I have had phenomenal mentors, although predominantly men, throughout my career. And I'm thinking about when that may have really pushed me to behave in a way that's different, because they brought a different perspective. So 2 little examples. When I was going for a fellowship position, I wanted to have a conversation about salary, and I was uncomfortable. I didn't know if I should or how I would do that and what it would come across like. And I had male mentors and friends talk me through it and support me in doing that. And I agonized over the email, but I did it because I felt I had enough support that I was confident. And when I was applying for tenure track positions, I didn't necessarily meet all the essential criteria and the job description. And I think there's evidence that many times women in our field or in our career type don't go up for jobs if you don't tick all the boxes in advance. And I had male mentors and friends and colleagues say, no, just apply, and encourage me and help me address these gaps. And I get the job, right? So I've identified that I do fall into some of these stereotypes that are out there, where maybe as a woman or maybe just as me, I really benefit from having some mentors who view things very differently to help me with some of these key moments in career progression. But I think what Jean and I have talked about is, just as you need those types of mentors, you also need mentors who are very similar and have very similar experiences and challenges and environmental factors. And you want that diversity of guidance and advice and support. And so some I can find naturally, and some right now, just because we're improving, but we're not as diverse as we should be at all levels, we need to maybe make extra effort to connect senior faculty, junior faculty, postdocs, all the way down, to make sure that all of these viewpoints are kind of contributing to some of the professional development challenges we have.
Yeah, I mean, it seems like, you spoke to the idea of having mentors, and I think you're right. I think that somehow mentors that sort of can really understand how you're thinking and anticipate your perspective, and having female mentors in that regard is I get what you're saying. If and I'd like to think my advice that I give to female and male postdocs is the same, but maybe it shouldn't be the same. Maybe it should try to anticipate, what's going through the mind of female postdocs, and sort of like in terms of, what they should ask for or not. I mean, it's similar, I think, but you're right. I mean, I think that there's probably advice that would be better given for female mentors potentially. So, and hopefully it's changing too. Hopefully, I look at this problem as sort of a systemic problem that's, we can only try to get rid of bias and foster, practical things like, or even transparency. And I was talking to Danny Bassett a couple of weeks ago, being aware of what is the teaching load of females versus males? What is the childcare sort of issues, and why can't we sort of help that?
So I guess in regards to OHBM, I know there is a mentorship program for postdocs, but maybe this could be extended into the group of junior PIs. I'm not saying just women PIs, but junior PIs in general. But I also want to stress that some of the biases that we may experience or others may experience, they may not be conscious. They're unconscious biases, and that's very much a part of our nature, and that's part of the world. And I think when I also really appreciate my mentors. All of my mentors have been great, but when I sometimes bring problems to my male mentors, and then they'll say, I've never had this problem. I don't know.
Right.
This might just be you.
Yeah.
And so it's an incredibly lonely feeling. And I think it doesn't have to be. That's sort of whether we organize ourselves locally or at the organizational level or internationally. I mean, seeking out mentors or having a mentorship structure or framework for this type of support, I think would help to get a lot more women adapted in academia, because I feel my challenges are not doing the work, but doing the other things that are not related to my training, not related to my science, and that can be understated a lot.
Yeah. Yeah, no, definitely. I see your point. And it seems, hopefully, I mean, it looks like you're spontaneously organizing to have this group, but I think, having something more formal in at least, I mean, in the context of OHBM, we could try to obviously more formalize that. And you're right. I mean, it's always interesting when I'm on committees at OHBM, you want to, there's a couple pools of people, one who just doesn't want to care even to look at the number of females being invited to talks or whatever. And there's another one that wants to maintain a quota. And then, there's just always this tension in that regard, which is too bad. I mean, you definitely want to build up more of a base of and have equal representation of female and male scientists. And, but I think until then, we have to really be mindful of the disparity of even things like who gives talks or that sort of thing, where it's even less of a, there it's sort of not like the subconscious bias. I was actually amazed with in references, that I don't even pay attention to who's an author or not typically. I mean, I just look at the paper and I'm like, oh, okay. But really, I mean, it's a large bias in male over female for references. And so a solution to that, and like you were all saying, like both of you are saying, is just making things transparent and then having clear organizations of females that could foster this mentorship, like you said, all the way down, all the way from the highest PI down to the student, and that really address the needs of females and try to solve some of the issues in that regard. So, yeah.
So personally, I want to contribute to this if I can in any way, through the OHBM, for example. Please let me know. And if anyone who's listening to this podcast, who's in this sort of situation, who would like to reach out for mentorship, I just want to pass it forward. I want to offer what I have.
Yeah. Okay.
And I think to everybody who's in a trainee position, something that I always experienced was super imposter syndrome, and I still have it, right? And I think a lot of people have it, yeah. But you do belong in the room, you do belong speaking at the podium, and you do belong being nominated for things or going up for things or applying for things. And sometimes you feel like you need an extra push. And so I would encourage, particularly for all women in science, nominate each other, support each other at going up for these things, introduce colleagues, build the networks. Because I think that there is a lingering feeling of uncertainty as to whether or not we're really supposed to be in the room for certain conversations. And we are, and it's definitely improving, but we can always do more. So I look forward to getting involved with all of these potential initiatives that might spring from this in the future.
Yeah, you mentioned imposter syndrome. It's so interesting. My wife is actually in the field of cardiology, and she was just hosting a meeting about this, talking about imposter syndrome. And I think, right. I mean, all people have it, but I think you're right. Females sort of have this sense a little bit more, it's like they definitely, and I think that should be all the more emphasized that, and I don't know, I'm not a therapist. I don't know how to, if someone comes up to me and says, I have imposter syndrome, I'm like, all I can say is, don't have imposter and try to assure them. But I'm sure that there's better ways of it addressing that. I think as we're advancing, there's always that hint of do I belong here? And yeah, I don't know the best way to give advice to that sort of thing. But I think there are people who do, and you're right. I think having groups that help people in this regard would make a huge impact, because I didn't realize this. My wife was talking about it, then all her female colleagues mention it, and I think it's a little bit with males, but not that much, and mostly with females. And yeah, maybe there's a lot of subtle cues, and there's a lot of things internally, and it all builds up. And how do you give advice? There needs to be professionals who I'm not to say, professionals who know best how to do this.
I think bringing in people who are trained in this is always a good idea, but I think it's also about, just exposure and environment from the very first moment you join a community. So with the earliest members just starting out in this field, can we introduce them to the scary senior people and make it a low stakes environment where you really can ask all the questions you have and feel like it's okay that you are junior, you don't know a lot yet, but you are welcome in this career direction. And so linking, because I have to say that I find and continue to find some of the biggest names in our field intimidating. And then I meet them and they're wonderfully supportive and nothing but friendly and generous with their time. And Peter, you're included in this. So I think it's enabling these one-on-one interactions between the newest members and the established members so that we can really get this off on the right foot. And then, yes, we need to maintain this throughout all the different career jumps that we all go through. But I would like to see that connection the moment we walk into the door.
Yeah, I agree. ISMRM has this great thing. One of the first nights they have a newbie session or something like that, or like a mentors, meet the experts. And so, you have some experts who are high up in the field and they're just standing at a table and people come up to them and they try to be as welcoming as possible. That's one thing that could be done.
Things like that are brilliant, I think.
Yeah. And just having it's interesting, especially with Zoom, you could actually have sessions where sort of some expert just, enters into something and then anyone could log in and just chat with them and talk with them or she or he. In that regard, I think you're right. That's part of that is to realize that, hey, that even people at the highest level don't have everything figured out by any means. I still get intimidated. Honestly, I have to admit, 2 weeks ago when I was interviewing Danny Bassett, I was like, oh shoot, I better know what I'm talking about. And also with you as well. I mean, obviously, but your expertise are more close to my expertise, but
and it's the awareness that some of these issues are going to maybe be more present in young women scientists or in anyone who doesn't feel represented across the discipline. So I like when initiatives like this are open to all, but remembering that sometimes it is helpful to say, like we're talking about today, maybe make some of these opportunities really focused for women. But it is going to benefit pretty much everybody, because I think there's work on mentoring and that we could do for all people in our community. But there are some nuances depending on who you might be trying to help.
Yeah. So, and maybe OHBM might be the best format for that. We should talk more, obviously, after this podcast. But also, I think there's a large push to do something solid and to actually have real meetings and real organizations in place. So.
So I absolutely would support that, and I would want to contribute. It's sort of like with kids, you have the big brothers and big sisters organizations, and so in academia, I feel like to be initiated. When I was a trainee, I shared that feeling with a lot of other trainees that, these are big names, senior people, and how do we get into their network? It's very daunting. So to have someone to initiate them will probably be helpful. To know that, although you, for example, have a big name, but you're also very friendly and approachable. I didn't know that really for the longest time.
No, okay.
And also we don't want to just be a quota. We want to. I think that just reinforces that imposter syndrome. Right. I'm just here because of the quota.
Yeah.
That is negative. I feel like we need to, be part of the conversation because we earned our spot at the table. I think that needs to be recognized more.
Yeah, I completely agree with that. Because having quotas like that, it sort of on some level fosters resentment in some people, and then still, right, like you said, it could potentially be a vicious circle, which fosters more imposter syndrome if you sense that they feel something weird about it. But just having it naturally happen, sort of, having, more support and more focused support on specific needs, or the various life of, definitely a junior female scientist has to, and all the barriers, it's not like it's a massive barrier like it used to be, but it's sort of like there's subtle things, and those build up, and that change the trajectory of people's careers. And if people have to work a little extra hard to overcome those. So having groups like that support those mechanisms I think would be really, really useful. So, all right. Well, hopefully this will catalyze something that will translate to some real action in OHBM. And I'll certainly, to the extent that I'm part of OHBM, I'll remind them of this, and hopefully they'll listen to this podcast. All right. We've gone over time a little bit, but thanks for staying on. So is there any advice or anything like that you want to give, or any parting thoughts with regard to this or even the previous things that we talked about? Yeah, especially with regard to what we're talking about here.
So I have one piece of advice, I think, that I've thought of all the advice that I've received over the years, and this one has really stuck with me. Basically, it goes something like this: just work on what you're interested in and passionate about, and don't be distracted by the chatter. So as I was starting my lab, I was given this piece of advice, because every day so many papers come out, people take different approaches, and one might not want to miss anything. So we might be hovering between different directions. And basically this is sort of saying, be confident in your ideas and pursue them. And I found that to stick to that piece of advice has really helped me over the years.
Yeah, and hopefully in a way that is in line with what you're saying. I think what I've found with my research and what I've seen from my mentors, and I'm trying to bring with my own trainees, is there are very real practical requirements to succeed and progress in academia. So within this direction that you're genuinely interested in, I think it's good to balance the small, mundane, incremental advances with the big, exciting, I really hope it works, this would be hugely impactful. I think balancing those two types of studies, it helps, you sort of pay the bills, everyone can eat, and you can reach for some of the most exciting research that you can. So I definitely echo, Jean, what you just said, which is find a direction that engages you, like genuinely interests you. And then I think it's the strategy of navigating, how can you make sure you're always progressing and occasionally able to make a great leap forward, right? So I think hopefully both of those things can happen. And then the methodology improves and our understanding of human physiology improves with this approach.
Yeah, that's really good advice. I think that, and it's really good advice in terms of, how to deal with the feeling that there's this onslaught of papers and people are being pulled, your ideas are being pulled all over the place. And I think I totally agree with both of you. Just finding something that you're passionate about and thinking deeply about it. And even at that stage, even thinking deeply about it just a little bit, you're already way ahead in that regard from most of the field, to sort of thinking about it in your own way and then trying things along that way and not be afraid to try things. So that's a great piece of advice. The opposite way, trying to triangulate between different papers and say, oh, well, they haven't done this, so maybe that's sort of the opposite way, as I think what works best, is just kind of, finding your questions that drive you. Yeah. Great. Well, this is really, really fun. Yeah, I had a whole bunch of other things to talk about, but maybe some other time when you come back or whatever. But thank you.
Happy to.
Yeah. All right. Well, good. We might have an ongoing discussion on physiologic fMRI. Maybe we'll have a big group or something, and maybe another discussion of maybe just a whole podcast devoted to women in neuroscience as well, which I think could be useful. So, but thanks again. I really appreciate you spending your time, and I think that's this raised the awareness of the community on a lot of things, from physiologic fMRI to women in neuroscience. So thank you. Thank you.
Thank you. It's been a pleasure.
Pleasure.
Great.